Welcome you all in CEC Gurukul online lecture. Learners, in previous class of series of lipids, we had discussed terpenes and biosynthesis of terpenes, which are derived lipids, and also we had learned their biosynthesis of squalene. Squalene, which is a biochemical intermediate. Also, we can say it is a biochemical precursor to all steroids. In this class, in this lecture, we will discuss lipid soluble vitamins, that are vitamin A, vitamin D and vitamin E and K are lipids soluble vitamins, and also they are the derived lipids. They are the lipids because they are soluble in fats. Learners, as we know, small amounts of vitamins are required in diet to promote growth and health. Vitamin A and vitamin D, vitamin E and K, you can see the structure of these vitamins on your screen. And vitamin A, this is also called retinol. Vitamin D, that is calciferol, and vitamin E, that is alpha tocopherol. So on your screen you can see the structure of these fat soluble vitamins, and these are the class of derived lipids because they are soluble in fats and absorbed and transported in similar way to that of fats. If we talk about the vitamin D is also fat soluble vitamin. Active form of vitamin D promotes intestinal absorption of calcium and phosphorus and influence bone mineralization. And vitamin D2, that is ergocalciferol, it is a commercially produced by ultraviolet or UV radiation of plant sterol, that is ergosterols. Similarly, vitamin D3 is formed by the action of sunlight on precursor 7-dehydrocholesterol in the skin. The human body utilizes both forms of vitamin D, that is D2 and D3. Similarly, you are looking on your screen the structure of vitamin E, that is alpha tocopherol. The vitamin E is an important antioxidant that is thought to protect polyunsaturated fatty acid from oxidative destruction in cell membrane. And similarly, vitamin K is also fat soluble vitamin, and it is needed in the liver for formation of blood clotting. And if we talk about because there are two types of vitamins, that is fat soluble vitamins and water soluble vitamins. So water soluble vitamins like vitamin C, it is also known as L-ascorbic acid, and this vitamin C is human are unable to biosynthesize this vitamin C, and it is a essential dietary components. So it is also involved in biosynthesis of protein metabolism, and also vitamin C is required for the biosynthesis of collagen, and it is also plays a vital role in wound healing, and also it is an important physiochemical antioxidants. So these are the vitamins we will discuss over here, especially vitamin E. So if we talk about the water soluble vitamins, example vitamin C. So vitamin C is water soluble vitamin. And why water soluble vitamins are essential vitamins? Because these vitamins are rapidly eliminated from body, and as a dietary levels needed to be relatively in high amount, right? So learners, here in this vitamin A, it is a lipid and it is a terpene, tetraterpene beta carotene, and we can synthesize that vitamin A, we can biosynthesize vitamin A from the beta carotene. Similarly, here in vitamin A, this vitamin A, which is a fat soluble vitamin, it is a group of fat soluble retinoids, also called fat soluble retinoids, and retinols, retinol and retinyl esters. So we can also call this vitamin A is a group of fat soluble retinoids, retinol, retinol and retinyl esters. And vitamin A is critical for vision, and it is an essential component of rhodopsin, which is a protein or sensory protein that absorb light in retinal receptors. So in milk, fat soluble vitamin A and D are relatively stable to heat, and it is a treatment used for the processing of milk. So the fat soluble vitamins are stored in adipose tissue and it is not directly absorbed with the bloodstream. Learners, this fat soluble vitamin, that is the vitamin A, it is biosynthesized from the beta carotene, which is a carotenoids, and in the previous lecture we had discussed about the carotenoids. What are the carotenoids? How can we biosynthesize the carotenoids? So this vitamin A is biosynthesized from the beta carotene, which is a carotenoid, and beta carotene is biosynthesized via common precursors isopentyl diphosphate and dimethylallyl diphosphate, which are biosynthesized by mevalonate pathways, which we had discussed in the previous lecture. So this beta carotene compounds gives yellow, orange and red coloring to the vegetables and fruits. And learners, our body cleaved beta carotene, which we get from the red, orange, yellow color vegetables and fruits, converted or cleaved into two molecules of vitamin A, and that vitamin A is responsible for the vision. So vitamin A is a precursor of rhodopsin. Rhodopsin is a complex pigment which contain sensory protein. So this biosynthesis of vitamin A from the beta carotene is most likely a dioxygenase reaction, where molecular oxygen react with two central carbon atom of beta carotene. So we will see here biosynthesis of how vitamin A is biosynthesized in our biological system. So that beta carotene or carotenoid, which is a terpene, it gives two molecules of retinol, and that retinol, reduction of that retinols to retinols takes place, and that newly formed retinol esterified with long chain fatty acids to form retinyl esters, and that retinyl esters taken up by liver, where vitamin A is stored in ester form in the body. So that vitamin A which is biosynthesized from the terpenes, that retinol is stored in the form of retinyl esters in our body. So here in the reaction you will see here how the vitamin A is biosynthesized. So this is the structure of, on your screen you can see here, this is the structure of beta carotene. So this is beta carotene, and as you know, beta carotene is a here contain isoprene unit. these are the isoprene unit you can see on your screen, and that beta carotenoids, it is here deoxygenase reactions, where molecular oxygen react with the two central atom of the carbon of beta carotene, which is followed by cleavage of central double bond of beta carotene, and the reaction takes place and to form two molecules of retinols. So here you can see here this beta carotene cleaved at the central carbon atom of the double bond with the reaction of molecular oxygen at two carbon atom, and the central double bond which is cleaved and to form two molecules of retinol. So in the next step you will see here biosynthesis. This biosynthesis takes place in intestinal mucosa, or in the structure B you can see that molecular oxygen on the central carbon atom to form a epoxide type ring, and then here the reduction takes place and we get retinol, and this retinol on reduction gives retinol. So reduction of retinol to retinol during the process of digestion takes place. So this is the biosynthesis of vitamin A, that is called retinol, and from where we get vitamin A or beta carotene. So as we had discussed in the previous class, the beta carotene from the food sources such as carrot and other colorful vegetables and fruits, which is red, orange colors, which are the precursors. These are the called beta carotene, and from that beta carotene is produced. So this beta carotene act as a precursor of retinol. Retinol and retinaldehyde in turn produces retinol, which act as a precursor of retinoic acids. So this retinol, retinoic acid is a vitamin A metabolite. In the next reaction you will see, this is the structure of, you are looking on your screen, that is the structure of retinol, where we have OH group in the end and a ring, that is that ring which contain a CH3 group. This is called beta ion ring. So that retinol exists as a provitamin A in vegetables, and it is stored as a retinyl fatty acid ester, which is in trans form, which is present in the trans form. So vitamin A has a special role in eye as key indicator of rod phototrans reductions. So this retinol is converted to retinal rods. Rods are the cells which are present in the eyes there, because there are two types of, that is rods and cones. So that retinol which we get from the beta carotene is converted to retinal in the rods to 11 cis retinol to 11 trans retinol, which is bound to opsin, and opsin is a protein, and that opsin which is bound to 11 cis retinol form a rhodopsin. So rhodopsin is a conjugated protein which is present in rods of our eyes. So that role of vitamin A in our eye takes place. What is the role of vitamin in our eyes? It is very important for our vision cycles. So because here, as I told you in the previous slides, that beta carotenoid which is present in red, orange and other colorful vegetables or fruits, here it that changes into retinol, and which is in turn esterified in the esterified, and which is stored in the liver, and when needed it change to retinol, and that retinol is bound to the proteins, and that enters into the retina, which is target cell in the rod cells, where it is oxidized to retinal, and that retinal complexed with the rhodopsin, complex with the opsin, that change into a conjugated protein which is present in rods of our eyes, that is rhodopsins. So that rhodopsins, from that rhodopsin we get photopigment of the vision. So that this is a rhodopsin, which is a conjugated proteins and which are present in our rods of our eyes, are responsible for the vision of our eyes. So here in next slides you can see the aldehyde group, that is retinal, which is linked to amino acid, E amino acid, that is lysine, and this lysine is from opsin, and retinol, which is primary alcohol and contain beta inon ring. Then this retinol, which is also called vitamin A, having 2 isoprenoid unit, 4 double bonds and 1 hydroxyl group present in that retinol, and it is present in animal tissue as a retinyl esters, and retinol and retinols are interconvertible. Retinol and retinols are interconvertible. So here that rhodopsin is a primary photoreceptor molecule of vision. So always remember, the vitamin A, it is released from the liver as retinol, and zinc is essential for retinol metabolism. So it is the structure of retinyl esters, and that retinyl ester hydrolase, this is also called REH, and also lecithin retinal acyl transferase, that is LRAT, that is in present in liver, and here that retinyl esters is stored in liver in our adipose tissues along with this retinol, this vitamin A. So here this rhodopsin, which is a primary photoreceptor molecule of vision, it contains opsin. In the next PPT you can see here, this is slides you can see here the all trans beta carotene. In presence of beta carotene monooxygenase that convert to trans retinal, so beta carotene to trans retinal, and then in presence of retinal dehydrogenase, it all trans retinoic acid, and then again all trans retinol, and finally retinyl esters. So it is a storage form which is having acyl chain. So this retinyl esters, which is stored in our liver or adipose tissues or in muscles, here contain that ester groups. So this is the structure of here retinol, which is having OH group, and that retinol, which is vitamin A, oxidized to aldehyde, which is having trans double bond at C11 carbon atom, and isomerized to cis double bond, that it convert into retinol, which is bonded to proteins, and it is called opsin, and that opsin uses as a lysine side chain. So in the structure you can see here there is a opsin, and this opsin bound with the that retinol. So this opsin uses as a lysine side chain, and it is having a imine with here retinal position, retinal C11 carbon atom. So this is called rhodopsin. So the rhodopsin is having two parts. One is opsin and other is a chromophore group. So in this structure you can see there is a activated rhodopsin. So one is having opsin and another is having chromophore. So this rhodopsin is responsible for the vision in our eyes. So rhodopsin, as I told you, it is a contain opsin molecule and chromophore, which is 11-cis retinol. So in the structure you also can see in the slide, this is activated rhodopsin. It contains opsin, and other side of the structure is chromophore, which is 11-cis retinol. So bleaching of visual pigment takes place. Here the trans isomers is not stable and it is hydrolyzed to 11E form, that is retinal and opsin. So it is also called bleaching of visual pigments. So learners, bleaching of visual pigments takes place when raw photopigments are exposed to light, and it is called bleaching because the photopigment colors becomes almost transparent. So here the 11E form of retinal is then converted back to 11Z forms or isomers. So retinal to complete visual cycle. This is the complete visual cycles where the geometry of that retinal pigments takes place. So here in the next PPT you will see here this is the this is the opsin, and that opsin which is having a chromophore side chain that convert into 11E retinals. So we can say here activated rhodopsins during the visual cycle geometry change of the molecules, and which is responsible for initiating the process of vision cycles. So this in the next slide you can see this is the vision cycle, and this vision cycle, and this is called Wald vision cycle, and it was elucidated by the George Wald, for which he received the Nobel Prize in 1967. This visual cycle is the biological conversion of a photon into an electrical signal in our retina. So the protein complex receptors which we had in the previous slides, that is called opsin, and that opsin contains chromophore and 11-cis retinol. So that 11-cis retinol is covalently linked to the opsin receptors, and isomerization and release from the opsin protein, and during that isomerization all trans retinal is reduced to retinol and travel back to retinal pigment, and due to that pigment to be recharged, and then it is esterified by lecithin retinol acyl transferase, that is called LRAT, and then it is converted into 11-cis retinol. So this is the complete visual cycle you can see on your screen, and this is also called Wald vision cycle, and within the eye 11-cis retinol which is bound to a protein that is called opsin, and that opsin form rhodopsin, and that rhodopsin present in our rods, and here another pigment that is iodopsin that is present in the cones, and at lysine residue all 11 cis retinols convert into the trans forms. So geometrical changes takes place during the vision process. So this is the complete visual cycle, and as I told you, the vitamin A, which is a fat soluble vitamin, and that is responsible for our vision. So the recommended dietary allowances for vitamin A expressed as retinol equivalents, that is RE. So 1 RE equal to 1 microgram, that is retinol, 6 newgram beta carotene, 12 newgram other carotenoids. So children recommended that daily allowances for vitamin A for children, it is 400 to 600 newgram per day, men's 700 to 1000, and women 750 newgrams per day. And the source of vitamins A are animal milk, butter, cream, eggs, or fish liver, egg or fish liver oil, and vegetables which contain yellow and orange pigments. So learners, in this class we had discussed fat soluble vitamins. What are the fat soluble vitamins, why they are in the class of lipids, and how can we biosynthesize the vitamin A. As we have seen there, vitamin A is biosynthesized from the carotenes, and that is the beta carotene. Beta carotene is a carotenoid which is a terpenes, that is a tetraterpenes, and that beta carotenoids convert into retinol, and that retinol which is bounded convert into retinyl esters, and which is stored in our liver or adipose tissues, and that retinol which is bounded to a protein that is called rhodopsin, and that rhodopsin is responsible for the vision cycle in our biological system. Thank you.